So as we move into chapter 4, we'll begin looking at whole cells. However, this is probably the largest we'll get all semester long, because as soon as we finish this chapter, we'll be diving right back inside the cell to look at some of the smaller processes that go on in there. Cells are characteristically microscopic in size. Although there are exceptions, the typical eukaryotic cell is about 10 to 100 micrometers. And the typical prokaryotic cell, that is the bacterial type of cells, are about 1/10 of that, 1 to 10 micrometers in diameter. Because cells are so small, they were not discovered until the invention of the microscope in the 17th century. Cells were first discovered by Robert Hooke in 1665. Another early microscopist was Leeuwenhoek. Leeuwenhoek first observed living cells, which he termed animalcules, or tiny little animals. After these very early efforts, a century and a half passed before biologists fully recognized the importance of cells. In 1838, Matthias Schleiden stated that all plants are aggregates of fully individualized, independent, and separate beings, namely the cells themselves. In 1839, Theodor Schwann reported all animal tissues also consist of individual cells. Thus, the cell theory was born. So what is cell theory? In its modern form, the cell theory includes only the following three principles: that all organisms are composed of cells, that cells are the smallest living things, and cells arise only from pre-existing cells. So in this sense, the cell theory is quite simple. All cells today represent a continuous line of descent from the first living cells that are said to have arose 3.6 billion years ago on an Earth very different from the Earth that we live on today. Now, most cells are relatively small, and this has to do with the idea that they have to be able to get substances in and out of the cells and serve all parts of the cells. So if the cell were to get very large, then it would take a very long time for substances to diffuse into the center of the cell. The rate of diffusion is going to be affected by the area of surface available, also the temperature, the concentration gradient, because if there's a lot of substance to diffuse into the cell from the outside, then more of it will. But if there's not very much, then much less of it will. And then distance is key. When we think about distance, it's a matter of how long is it going to take for materials to enter the cell here and reach the center of the cell in here. In a larger size cell, this distance is much further, and thus the rate of diffusion alone of substances is not enough. So it's because of these factors that cell size is limited. If we look at the table down here, you can see that if we increase the radius by 10 times, the surface area to volume ratio becomes much, much, much less, such that at a certain point, the cell would not receive as much nutrient as it needed in the center if it became too large. That is, that as a cell's size increases, its volume increases much more rapidly than its surface area. Some cells overcome this by becoming long and skinny, like muscle cells or neurons. Organisms made of many cells have an advantage over those organisms that are composed of fewer larger cells, because each individual cell has much more likelihood of getting the nutrients that it needs and ridding itself of waste in an appropriate time. So not many cells are visible to the naked eye. Most of them are less than 50 micrometers in diameter. So to visualize a cell, we need the aid of technology, and the development of microscopes and their refinement over the centuries have allowed us to continually explore cells in greater and greater detail. The reason that we cannot see such small objects is the limited resolution of the human eye. Resolution is the minimum distance that 2 points can be apart and still be distinguished as two separate points. When two objects are closer than about 100 micrometers, the light reflected from them strikes the same photoreceptor cell near the rear of our eye. Only when the objects are further apart than 100 micrometers can the light from each strike different cells, allowing our eyes to recognize these as two separate objects rather than one. By using a microscope, we can magnify the object, and thus our eye can see the two objects as much farther apart and resolve them. So there are two basic types of microscopes. There are light microscopes and electron microscopes. Modern light microscopes operate with visible light. They use two magnifying lenses to achieve a high magnification and clarity. The first lens will focus the image of the object, and the second lens magnifies that image again and focuses it on the back of the eye. Microscopes that magnify in stages using several lenses are called compound microscopes. So although the light microscope can resolve structures that are 200 nanometers apart, so not micrometers, but nanometers, we have a limit to the resolution we can achieve using light rays because of their wavelengths. One way to overcome the limitations of light are by using an electron microscope, because the wavelengths of electrons are shorter. An electron microscope employs electron beams and has 1,000 times the resolving power of a light microscope. There are two types of electron microscopes: transmission electron microscopes and scanning electron microscopes. Transmission electron microscopes are so called because the electrons used to visualize the specimen are transmitted through the material. They're capable of resolving objects only 2 nanometers apart, which is only twice the diameter of a hydrogen atom. The second kind, the scanning electron microscope, the beams of electrons are shone on the surface of the specimen, and the electrons are reflected back from the surface, together with other electrons that the specimen itself emits as a result of the bombardment. These are amplified and transmitted to the screen. You can see a variety of images over here on the right using different types of microscopes. You'll notice that the ones with electron microscopes are very, very clear. When cells were visualized with microscopes, there were two basic cellular architectures that were recognized. There were eukaryotic and prokaryotic cells. And this refers basically to whether a cell has a nucleus or not, the karyo being the nucleus. So the prokaryotes are the very simplest of organisms. They lack a membrane-bound nucleus. However, they do have DNA, and it's present in a nucleoid region within the cell. In addition to having a plasma membrane, prokaryotic cells also have a cell wall outside of the plasma membrane. Prokaryotic cells lack membrane-bound organelles. However, they do contain ribosomes. Eukaryotic cells also have ribosomes, but the ribosomes are of a different type. They do perform the same functions, but they're definitely of different origin. There are two main types of prokaryotes. They fall under the domains of archaea and bacteria. Now, bacteria are the common prokaryotes that you're familiar with. However, archaea are said to be the extreme bacteria. Although they are very similar in size and structure to bacteria, they live in extreme environments. You might see thermoacidophiles in some of the sulfur hot springs. So they live in extreme hot, extremely acidic environments. Whereas bacteria are the ones that coexist in the sort of environments that we live in. Let's take a quick look at prokaryotic versus eukaryotic cells. First of all, we've already learned that prokaryotic cells are about 1/10 the size of eukaryotic cells, but they do have some basic structural similarities. They each have a nucleoid or nucleus where the DNA is located. The prokaryotic cells have a nucleoid region and no membrane-bound nucleus, whereas eukaryotes have a nucleoid region held within a membrane-bound nucleus. Both cells have cytoplasm, which is the fluid or semi-fluid matrix inside the cell, which contains the organelles and the cytosol, the liquid itself. Both prokaryotes and eukaryotes contain ribosomes, which is really important because ribosomes are necessary for the synthesis of proteins. The instructions on the DNA are translated into proteins by the ribosomes. And each prokaryotic and eukaryotic cells have a plasma membrane, which is composed of a phospholipid bilayer. We'll learn a lot more about the phospholipid bilayer in the next chapter. Prokaryotic cells have a cell wall outside of that plasma membrane. So bacterial cell walls are composed of peptidoglycan, whereas the cell walls of plants and fungi are composed of different sorts of materials. But peptidoglycan is what you need to remember about bacterial cell walls. They protect the cell and maintain its shape, as well as preventing excessive uptake or loss of water. Both bacteria and archaea have these cell walls, although they're slightly different. The susceptibility of bacteria to antibiotics often depends on their structure of their cell walls. For example, the drugs penicillin and vancomycin interfere with the ability of bacteria to cross-link the peptides in their peptidoglycan cell wall. So this is like removing all the nails from a wooden house. It sort of destroys the integrity of the structural matrix, and so the house no longer stays together. Basically, this makes the bacterial cell wall porous to water, so it rushes in and swells the cell and causes it to burst. Now, we're still learning the physiology of the archaeans. Most of these organisms are difficult to culture in the lab because of their extreme environments. And so this group has not yet been studied in a whole lot of detail. We do know that the cell walls of the archaea are composed of various chemical compounds, including polysaccharides and proteins, but lacking the peptidoglycan component. We also know that their membrane structure is different. And some of these features that are different in the membrane structure confer greater thermal stability to the archaeal membranes, allowing them to live in their very extreme environments. Some prokaryotes move by means of flagella or little tails. They're long thread-like structures. They may have one or more, or they may not have any at all. They use them in a rotary motion to propel the cell throughout its environment. So now we've finished off our section on prokaryotes. The prokaryotes are very simple. Take a moment here to write down all the details that you remember inside a prokaryotic cell. In the next section, we'll move on to look at eukaryotic cells.